Hello, everyone. Welcome and thank you for joining us. Happy Friday to everyone. Today's project talk will be recorded and the video will be posted on the InnovATEBIO website by the end of next week. My name is Deanna Rupa, and I am the program manager with the InnovATEBIO grant at Austin Community College in Austin, Texas. I am your host today. Before we begin, I have a few housekeeping announcements. During today's presentation, please feel free to use the chat box to jot down questions. And additional information on today's ATE project is located on the InnovATEBIO website, which I will drop the link in the chat. And then lastly, we respectfully request that you complete a survey at the end of the presentation, which will help us in planning future events. Thank you. At this time, it is with great pleasure that I introduce our Project Talk speakers, Dr. Dave Micklos, the project PI and founder and executive director of the DNA Learning Center at Cold Springs Harbor Laboratory in New York, and Dr. Anna Feitzinger, the assistant director at the Science DNA Learning Center. And the title of their presentation is Fulfilling the Promise of Anytime, Anywhere DNA Sequencing. Welcome, Dave and Anna.
Thanks, Dion.
Yeah.
I was initially on this talk, and then I was off the talk, and Anna was on it, and now I'm back on to do a tag team match with Anna. I'm in Germany right now. I thought I might be traveling, but I'm here in this nice hotel in Dusseldorf. Anyway, there's a dead part of the talk, and there's a live part of the talk. And guess which part I'm giving? I'm giving the dead part, and Anna's giving the live part. But hopefully, you'll find something lively from what I'll tell you. I'm going to give the background to this whole project about our ability to sequence and analyze DNA just about anytime, anywhere. And then Anna's going to give you a demo of a brand new interface that we've been working on over the last year or so. So I'll share my screen here and we'll get started. As some of you know, I like to know where things came from. And I think as teachers, you probably do too. So I'll be telling you, how we got to where we are today with this project. This is Cold Spring Harbor Laboratory, and this is the DNA Learning Center in the village of Cold Spring Harbor. I'm not going to belabor that, but in case you didn't know, Cold Spring Harbor Laboratory has been home to 8 Nobel Prize winners. We sort of call them our own. And let's just see if you know what all of them did. A little quick test. All of you should get 100%. Al Hershey, Max Delbruck, and Salvador Luria worked with bacteriophages. And of course, Al Hershey did with Martha Chase, the famous blender experiment, which was the conclusive evidence after Oswald Avery that DNA is the molecule of heredity. Jim Watson was part of the team that discovered the structure of DNA. It's unfortunate his career's has gone downhill in the last few years. We can't get into that. Barbara McClintock discovered transposons or jumping genes. If there was ever a genius that lived at Cold Spring Harbor, she was certainly one. This might be a stumper, but this is Rich Roberts and Phil Sharp. They discovered RNA splicing or split genes, the phenomena of how the eukaryotic genes are made up, which we all know, but maybe wonder where that came from. And Carol Greider, who discovered telomerase, the enzyme that remakes the telomeres or the ends of the chromosomes. Going back in time, if you think about the revolution that many of us have been involved with, which is, involves these little gel electrophoresis chambers, prior to that revolution, it was very difficult to separate gene-sized pieces of DNA. It would have taken this analytical ultracentrifuge, which would have set you back about $500,000 in the 1950s. So it meant that initially to be in the DNA or the gene club, you really had to be at a research one and well-endowed institution. But when gel electrophoresis was discovered in 1973, it revolutionized the study of gene-sized pieces of DNA and really made it egalitarian because just about anybody could get into the gene club at that time. It revolutionized education too because, oh, and by the way, this was the paper that brought that about. It was published at Cold Spring Harbor, and Phil Sharp was one of the authors of that paper before he was a Nobel laureate. But my point here is that within a decade of the discovery of the structure of analytical agarose gel electrophoresis, we and other people, and probably you, were doing it in classrooms with high school and college kids. So that was truly a revolution, not only in the way we looked at DNA, but in the way we taught about DNA. And I'm happy that many of us here, were somewhat involved with that first revolution in DNA education. Well, this is the second one, you could say, which is moving from this kind of sequencer that was used in the initial Human Genome Project to this little miniature MinION sequencer, which Anna's going to be showing you and telling you a lot about. So this device really has made the sequencing of DNA egalitarian in the same way that the gel electrophoresis chamber made the analysis of gene-sized pieces of DNA. You've all seen this, but it still makes me excited to see it. For a long time, initially, the sequencing of DNA was ramping down in cost at about the same rate as the cost of putting transistors on chips. And Moore's law is that so-called law that determines how often does it take to double the number of transistors you can fit on a chip, and as you double the number of transistors, you decrease the price by about half. And you can see that the cost of DNA sequencing was more or less following Moore's law until about 2008, and then it dropped precipitously. And if you never did the math, we're hovering around here still, but that's 100,000-fold drop in the cost of DNA sequencing over a matter of less than a decade. So that's a real revolution, and we all sort of know that in our guts. About the same time that the price of DNA sequencing was dropping exponentially, we had a lot of websites being developed where just about anyone could go with and put their DNA sequences or grab DNA sequences and analyze them. So this was another revolution in biology coming along. And I've said this, I started saying this about that time. So it's been, unfortunately, about 15 years that I've been saying this, that it's the first time in the history of our field, biology, that students and teachers can work with the same data at the same time and using the same tools as Research One biologists. It's unprecedented. And we just need to remember that we still are in an unprecedented time in our field. Well, around this time when the cost of DNA was dropping precipitously, I became involved in an NSF project called the iPlant Collaborative. This is just a little bit about it, but its objective was to make a discovery environment, a cyber infrastructure to help biologists, especially plant biologists, in this, especially this new world that involved DNA and RNA data, as well as other kind of data. The DNA Learning Center was involved in it on the educational side, and our task was to develop a sort of educational tool to make bioinformatics easier for students and teachers. And we came up with, we worked with about 25 different collaborators from 11 different institutions, and we came up with this tool called DNA Subway. And, we used to say this used to be the way you could walk around bioinformatics. And what we wanted to do was make it so that students could ride into bioinformatics. And so we made this analogy to a subway. If you don't have a subway nearby, you can just think of trains and stations that work just as well. This just shows you that, starting out from zero in 2009, we've risen up to lots of sessions per year, lots of users registering each year. And it's a pretty going thing, and it even functioned very well even during COVID. Just some facts and figures about DNA Subway. I don't expect you to read all of these, but the main thing I want you to see that there's been approaching 1 million user sessions since we started this thing. Uh, over 250,000 DNA sequences have been entered into the database, usually by students and teachers doing projects. People say that it's easy to use. Um, we get a mixture of students from high school, 2-year colleges, and 4-year colleges. And I think the key thing in all of this is that when we've done pop-up surveys, which are still going now, the number's up to about 16,000 surveys have been launched by people who were using the tool. And the key thing is that most of the people who are there, mainly students, are doing independent or long-term research projects, and most of them are using their own data that they brought to the tool. So that's what we really were hoping with this tool. Now I'd like to shift gears and tell you about the other part that Anna will be talking about. We're going to be talking about DNA sequencing and analysis, but we're going to be talking a little bit about DNA barcoding as one use case in DNA sequence analysis. And I'm happy to say that DNA barcoding as a sort of sub-discipline also started at my institution, Cold Spring Harbor Laboratory, at a series of meetings held at our Banbury Center in 2003. And these are all the more or less founders of that little field of DNA barcoding. This is Paul Hebert, who's sort of the father of it all. And Mark Stokel, who's at Rockefeller University, he figures in the next step of this whole mess that I'm telling you about. Because this is just to give you an idea of a barcode. It really is like a universal product code. These are DNA barcodes here, and you can just see exactly how similar they are to this universal or UPC code. This is a mud crab that a lot of kids in our neighborhood were collecting for a while. And this is the barcode of these mud crabs. You can see they're all identical except for these little polymorphisms. But in the database, the most closely related other mud crabs had a totally different set of bars on either side. And these are relatives on either side of that mud crab. Well, Mark Stokel, and I'm watching my time here really carefully. Mark Stokel, who was at those meetings at Cold Spring Harbor, had a daughter named Kate, and she needed a project to do in high school. So her and her friend decided to do some DNA barcoding. And they went around to restaurants in Manhattan and collected sushi, which, isn't cooked and is pretty easy to do DNA barcodes on. The unfortunate thing, I hate to burst everybody's bubble, but they actually didn't do any of the manipulations. They didn't isolate any of the DNA or do any of the barcode amplifications or anything. That was all done at the Museum of Natural History. But the girls did actually collect these samples in the restaurants. And what they found out is that something like 40% of the samples were mislabeled. It was a cheaper fish being sold as a more expensive one. And these studies have been held up all around the world, where somewhere between 30 and 60% of the seafood that you get in a restaurant or around the markets is mislabeled. So that's sort of a fraud thing. We started doing DNA barcoding in 2010 and 2011. And since that time, we've run 3 different projects, related projects for students in the New York metro area. And this is just some facts and figures on the number of students and so forth. You don't need to memorize any of that, but let's just say we've done projects with about 4,000 students now. We have 2 different annual symposia where the kids get together and present their posters and hear a nice talk. One is held at Cold Spring Harbor and one is in New York City. And here's one of the posters early on, and I'm going to give you just about 3 examples of what kids can do with DNA barcoding. If your appetite has never been whet for it, perhaps I can whet it now. These were 3 kids from a little science high school in the Bronx. Not the Bronx High School of Science, but the Hostis Lincoln Academy, the little science high school that could. And here was their project. They went around New York City and collected ginkgo products. And they were smart. They mainly just did dry products. They didn't do tinctures or oily things. And these are perfectly easy to barcode. I just have to move this because I'm a little confused. Okay. And these were their different samples here. And here are their samples, and here are the barcodes. Now, the thing you need to know about a barcode, if two things are really closely related, they will not have any differences in their barcodes. In other words, two barcodes that are identical will just look gray like this. So what's striking about this is that when you look at the ginkgo barcode from GenBank, this or this one has a lot of bars here. And if you look at all of their samples, there's virtually no bars and none that seem to match any ginkgo ones, except not even that one. And these down here are sort of artifacts. But if you look at this big gray area and you look for something in a database that matches pretty closely, you actually come up with a good match to all of their samples. And that's rice. And we gave them the first prize in this competition because we liked that result, but we never reproduced it. We should have, because it was interesting, but I didn't know whether to believe it. But that experiment was produced about 3 years later. Now I got to get this to advance. Oh gosh, the advancing thing's not working. Oh, there we go. And this was the fellow who decided to reproduce that experiment. He didn't do it, the experiments himself, but he ordered it. It was the Attorney General of the state of New York. And they subpoenaed ginkgo products from Walmart, Walgreens, General Nutrition, and several other stores. And the same result was found for all of them as it was found for Walmart, which was they didn't find any ginkgo DNA in any of their products. All they could find was rice or ariza, exactly what the kids had done from Lincoln Hospice Academy. So, these food fraud things, which kids love, actually can be pretty interesting. And, this was an interesting result. Now, if you're trying to think of things, that make sense biologically that kids can do, because obviously kids will do a lot of experiments that you think are pretty crazy. And some of them actually turn out to be super interesting, although you thought they were crazy. But this is one that's easy to do, which is to barcode insects, such as this mosquito here, which carries some equine diseases. It usually isn't a problem for humans, but does carry some encephalitis and things like that. And what you'll find is here, these are all the records that were available at the time that this experiment was done in about 2012, I think. 2000, what's the date up there? I can't see it. Oh, 2019. These were the GenBank records that existed from other experiments, and here's the collection of a student up on Long Island. And what you'll see is that this is about 50 miles north of the most northerly expected range of this mosquito. And this is the perfect example and the perfect expectation you would have for global climate change, where a southerly species is increasing its range to the north. And we've shown this students have shown this with ants as well. And I'm sure there's lots of this experiment could easily be reproduced with other insects. You can also do publications. This was one we did of a little bio blitz that was done in a park in Brooklyn, and has 25 student and teacher authors. So that can be done. We started submitting GenBank records done by students very early on. It's probably the simplest publication that a student could ever do that actually contributes, a block to the edifice of science. And here was our first paper. And since that time, you can see we've had over 2,000 GenBank publications. And the key thing is here, over 1,600 different authors, and the vast majority of them are students and teachers. The last couple of things I want to leave you with is, many people believe that DNA barcoding is an especially good topic for course-based undergraduate research. And we've certainly been selling that and been big proponents of that from day one. Great thing about it is students can do lots of different kinds of projects. They can be individual ones, or they can be distributed projects where different students in different places or different students in different courses can work on the same project. But the key thing is you can do lots of different things, but use the same technology, the same infrastructure, the same biochemistry, and the same bioinformatics. So it actually is possible for one teacher to administer a lot of different independent student projects. How big can it get? Well, this is the biggest barcoding cure in the world, I'm sure, and possibly the biggest what I would call scaffolded cure, where kids are using the same method. This is at James Madison University in Virginia, still going strong, but you can see 800 students per semester are doing barcoding projects. Here's one of the lecture sections, which is just frightening to look at. And you can see there's 30 plus lab sections, and they have 20 different instructors that have all learned how to do this technology in synchrony. And if you like to know how to do that, there is a course source paper done by the JMU people that will tell you exactly how to reproduce that large scale cure that uses pretty much the methods that we developed at or popularized at Cold Spring Harbor, plus DNA Subway. And the last couple slides, and I'm going to turn it over to Anna in just 3 minutes. We're doing this work over the last 5 years or so as part of InnovATEBIO, which is sponsoring this talk. We're the genomics hub. We have a little hub system of InnovATEBIO where we have a distributed system of different groups and people with expertise that can lend their help to community college and other educators all around the country. We're based in New York City at our DNA Learning Center in Brooklyn. And, these are some of the things we do. But the key thing is we're really trying to help teachers with emerging technologies, helping them to implement cures, and just trying to also at our own center be a sort of model for best practices in ATE. I'll bring you to the last strand of this talk. We've talked about DNA Subway and DNA sequencing, and now the last strand is nanopore sequencing. We have been playing with nanopore sequencing for a number of years, especially Jason Williams in our group. But, the technology wasn't quite settled. There was a high error rate. And I would really just say in the last couple of years, it's really settled down into a reproducible technology that I would say is ready for prime time in teaching. And in 2023, Cold Spring Harbor Laboratory entered into a memorandum of understanding with Oxford Nanopore Technologies, which is the manufacturer of the most widely used nanopore devices. So, our job and our collaboration with Oxford Nanopore specifically is to popularize the use of these small sequencers in education and to make them cost-effective in teaching. We had a meeting about this last September where we really thought hard about the promise of nanopore sequencing in education. There's plenty of existing audiences besides DNA barcoding. There's a lot of people that sequence bacteriophages through the C-phages project of Howard Hughes. There's a lot of people who work with looking for antibiotic-producing bacteria with Tiny Earth out of the University of Wisconsin. And we heard lots of interesting projects already being done with Oxford Nanopore. And finally, this is why we brought you here today, is because I really think that we're at a special moment in biology education where we can take this little MinION device and couple it to analytics that will play well even on your telephone. And it's going to show you DNA Subway 2.0, which was really developed expressly, well, a couple reasons, but really expressly to merge with this nanopore sequencing technology and make this sort of anytime, anywhere, anyone DNA sequencing possible. And so I participated in that first revolution in biology education where we brought gel electrophoresis chambers into classrooms all over. And now it's not hyperbole to say that millions and tens of millions of students are now using electrophoresis chambers in high schools and colleges all around the country. People are using PCR machines to amplify different pieces of DNA. That was a great technology. And my belief is that in the next 5 years, there should be a nanopore MinION sequencer wherever there is a PCR machine and a gel electrophoresis chamber. So with that, I'll turn it over to Anna, who will show you DNA Subway 2.0. And if you have questions, maybe just save them till after Anna, because probably you'll have even more after she goes. So Anna, take it away.
Thanks, Dave.
Oh, I have to stop sharing. There you go.
Kick you off. Okay, there we go. Okay. All right. Can everybody see my slides, or is it the correct view? Or is
Yes.
it okay, great. I do have a few slides before I do the demo. So bear with me before we get to the live part. Just to backtrack a little bit, we do have a website called DNA Barcoding 101 that has all of the resources or the DNA extraction protocols, primer sequences, PCR programs that we use for DNA barcoding. So this is a really, really awesome resource that has all the information you would need if you were to want to do a DNA barcoding experiment with your students. We also have information on all of the projects. Dave had mentioned that we have these 3 programs for high school students, and that culminates in an annual symposium. All of their posters and projects are also on the website, and this is a really good source of ideas to see how other students or faculty have been using DNA barcoding with students. For nanopore sequencing, I am going to talk about how nanopore sequencing works in just a minute, but I did want to talk about resources for using the nanopore sequencer. In collaboration with a number of faculty, we put together a website, which is an ebook for a nanopore ebook for educators, and that contains protocols for using the nanopore for DNA barcoding, for 16S metabarcoding, and for phage genomics. This is going live on Monday, so we don't have the link yet, but we can send that around as soon as that goes live. Okay, so DNA Subway. Dave showed, I think it briefly, this is the old DNA Subway. It's more than a decade old. It looks like a subway, and the original DNA Subway had a number of bioinformatic workflows: a red, a yellow, a green, a blue, and a purple line. Our most popular and most used line is the blue line, which is the DNA barcoding line. Originally only meant to work with Sanger sequencing data. Because that is the most used line, that's the line that we have been putting the most time into developing for DNA Subway 2.0. But we will be working next on the purple line. We are working to make our bioinformatics workflows compatible with nanopore sequencing data. So I just had mentioned the barcoding line. That's going to be compatible with nanopore, and you're going to see that in just a minute. The purple line for metabarcoding, and also we're going to have a line to do small genome assembly. We talked a lot about DNA barcoding, but I just do want to mention that it is possible to do other types of sequencing experiments with students. Jason Williams and I have been teaching a summer camp for high school students for the past few years, where we've sequenced whole genomes the first 2 years we did plant genomes. The third year we did chloroplasts, and then this last year we did whole bacterial genomes. Students can use the sequencer to do 16S metabarcoding. So I taught a class looking at a waterway in New York City and looking at the distribution of different bacteria along that waterway with students. And for whole genomes, there are a lot of projects that can be done with bacteriophages, for instance. We hosted a course where students sequenced bacteriophages from wastewater. This was in collaboration with 2 community colleges in New York City. But why do we actually care about nanopore sequencing so much? This is because out of all of the sequencing technologies that are available today, I think this group would be familiar with Sanger and Illumina sequencing. Nanopore is the only technology that is portable. So I have a sequencer right here. If you can see my screen, it is very small. So you can actually work with students in the classroom to load it, to prepare the DNA. And it has the lowest startup cost. So the cost has changed over time, but it's a few $1,000s to start up. And we'll talk a little bit more about costs at the end. So the idea is the new DNA subway will be able to process both Sanger sequencing and nanopore sequencing. So in terms of, or in relation to DNA barcoding, the same extraction methods can be used, the same PCRs can be used, but our new DNA subway will be able to process different sequencing types. How does this nanopore sequencing work? So the nanopore in nanopore sequencing refers to a protein pore, which is this nanopore that is embedded in a membrane of the flow cell. So the flow cell is what contains the membrane with many, many, many of these nanopores. And DNA, which gets hooked up to this motor protein, gets shunted through the pore. And as the DNA goes through the pore, the current across that pore is disturbed. So what the device is actually measuring is the current change across the nanopore over time as the DNA goes through the pore. So just to go through that one more time, the library preparation is all about getting the motor protein onto the ends of the DNA. That library prepped DNA gets loaded onto the flow cell and can go through the pore. As the DNA goes through the pore, the current change is recorded across the pore. And the raw output, the raw data that is collected is the current change over time. That current change over time you can visualize as these squiggles, and that's what the company also calls it. And these squiggles need to get what's called base called into the DNA sequence. And that can be a very computationally heavy process. But just to get an idea of what this looks like, a squiggle actually it contains data for 4 base pairs. So it's not like a 1 peak per base. And this is a function of the dimensions of the pore. What you need to actually get started with nanopore sequencing is a computer to run the sequencer, a flow cell. This is what we load the DNA onto, and then library preparations to prepare the DNA with the motor protein. There's 2 major chemistry types. One is using ligases to essentially attach the motor protein, and the other method is called a rapid method, which uses a transposon complex, which simultaneously cuts the DNA and puts on the sequencing adapter. And these chemistries have unique indices that are put onto the DNA. So if you have 20 students that are working and each one has a DNA sample, their DNA sample will get indexed with a unique sequence so that it can be demultiplexed later on. For DNA barcoding, we found that this rapid kit works very well. So just from the PCR product, we can, with the students, do the rapid library kit preparation. And then all of the DNA gets pooled and sequenced at once. We use the Flongle, which is a smaller, less expensive flow cell. And I don't want to, yeah, okay, I'm okay with time. So with Sanger sequencing, we are so used to seeing this electropherogram. It's very easy to tell what's going on. There's a fluorescent peak that corresponds to the identity of the base. These are .abi files. When nanopore sequencing, this is high throughput DNA sequencing. And this can be a little bit different and challenging to describe to students. So one of the great features of the DNA Subway 2.0 nanopore line is being able to break this down for students and visualize it. And I'll show you how in just a minute. So the current change over time, that raw output file gets base called or turned into the ATCs and Gs, which are in what are called FASTQ files, which are similar to FASTA files, but they also have quality scores associated with them. And when you're running a nanopore sequencer on the computer, it generates a folder that looks like this. In this FASTQ pass folder is a folder for each sample. So the demultiplexing happens automatically as you're sequencing. So you can imagine if you have 18 students, as you're sequencing, each student will have a folder that is getting filled with these sequence files, these FASTQ files that contain many DNA sequences. Okay, so I'm about to do the demo. I just want to describe the data set that I'm going to be doing the demo with. This is a little bit of a contrived sample data set for the demo. Excuse me. But the source of it is an educator here at the DNA Learning Center, who's an entomologist, Jeffrey Petraka. He developed a forensic entomology lab. So the idea is using the identity of insects found on a cadaver to estimate their post-mortem interval. And he actually was able to acquire insects from the Forensic Anthropology Center at the University of Tennessee in Knoxville. And so with his students, they did DNA barcoding to determine which insects were on this cadaver to give an idea of the post-mortem interval. Okay, so I'm going to switch over to DNA Subway. So I just need to stop sharing for a second and share the web browser. Okay. This is under development currently. Can everybody see the DNA Subway? Okay, great. This is under development currently, so this isn't live yet. DNA Subway 1.0 is sunsetting June 30th. So by June 30th, this DNA Subway 2.0 website will be up and running. But right now, you can't, if you try to go to this website here, you wouldn't be able to use it just yet. But this is, it's undergoing alpha testing, so it might look different by the time it goes live. But so this is sort of our working alpha test. Okay, so I'm going to go into create project, and this is what the new blue line looks like. I will mention that we designed this to be mobile first, so everything, if it gets kind of condensed and squished on a phone screen, everything is meant to be able to stack and be able to be used on a phone fairly well. I'm gonna create a title, crime lab. And then as you can see, there's two options here, Sanger sequencing and nanopore sequencing. I'm just gonna demo a nanopore sequencing analysis. Oh, that already exists. Okay. All right. So the first thing we would want to do is to upload the files. I showed a screenshot of what that would look like, so you could get an idea of what that looks like when you do nanopore sequencing on your computer. So you could use this upload button to upload. I don't think you can see the pop-ups, but I would be able to upload that FASTQ folder that contains all the students' or students' DNA sequencing files directly to the website. I am going to use a data set that we already have in the backend, but this is available to upload the data directly from your computer. These are insects, so I'm going to choose CO cytochrome oxidase 1, which is the DNA barcode region for animals. And then the crime lab subset is data that was generated from Jeff's class that I had just described. I'm going to add that to project. And this shows the files. So these are the folder names that contain the FASTQ files that were generated from the student sequencing. We can't do much with them yet. So this is our edit window, and this is what we're going to do to filter out low quality reads. We're going to remove adapter sequences. I had mentioned that in the library preparation, we put on unique indices on the ends of the DNA, but we want to remove those from the DNA sequence. And then lastly, we want to take all of the DNA sequences and build a consensus, because we sequenced an amplicon. So we just really want to get to that one consensus sequence for each organism that we amplify DNA from. So these are the three tools. The tool for filtering low quality reads is called FASTP. So I can click FASTP all, and that will run FASTP to eliminate low quality reads for all the samples. Port chop is the name of the tool that will remove the indices. So I'm just going to kind of run them all, and then I'll show you what the output looks like for each of these tools. And then the last tool is called Medaka, and this is the bioinformatics tool that will take all of the reads or DNA sequences and make a consensus sequence from them. Okay, so I'm going to just show the output for FASTP here. This was a very, very quick run. I didn't mention that that Jeff had actually initially sent these for Sanger sequencing, but they didn't come back in time. So the day of with the students, when he realized he wouldn't have them, he just loaded it on the sequencer. So this was probably run for about an hour. So there's about 400 something reads, 427 reads. If you left the sequencing overnight, you would have tens of thousands of reads potentially. But for a DNA barcode for an amplicon of about 600 base pairs, it's really not necessary to have all that sequencing. So this is perfectly good. Okay, so before filtering, there are about 427 sequences. After filtering, there's 320, and what this is doing is just taking reads that have a Q or Fred score of below 9 and just eliminating them from the file. We do plan on eventually having the ability to change that Q score so that students could see how changing the Q score might change how many reads are taken out of a sample. But this is the FASTP report, and that is for each one. So I'm not going to go through each one. Port Shop, this is a command line tool. So we are still working on how we're going to present this on the website. But the important thing here is that this algorithm went through the sequences and found the indices, which corresponds to the index 4, which was, this is the sample 4. So it removed those sequences. And then Medaka, this shows an alignment of all of the sequencing reads to the consensus that was made. So Medaka generated a consensus. We'll look at that in a second, but the students can actually see the DNA read, the individual reads. So they're all stacked here. And how they align to the consensus. So where it's gray, it means that it matches to the consensus and all of the reads are in agreement. There are some single nucleotide deletions and some, you'll see some of these differences. This is most likely a sequencing error. I have to take a look at how the sequencing for this particular data set actually went. Sometimes some runs are not the most ideal, but for this one, you'll see that you can see where there are errors. And this is a really nice way for students to see the reads, just like you look at an electropherogram and say, oh, you know when you see a bad electropherogram because there's so many overlapping peaks or very short peaks. Looking at this, students can get an idea of that as well, visually.
Anna,
Yes.
I'm going to interrupt you just for a second. For those of you that remember the days or have ever taught about assembling a genome where you have to take all of the reads that are done of the genome and then align them to make a sequence, and then you'll know that they'll say that this genome was done at 100x or 50x or 150x. That means how many sequences there were at any given point. So this genome, although there's fragments, I don't know what the depth is, but it's probably 100x. But this is analogous to when you would assemble a big chromosome or a genome by aligning the reads. That's all. Sorry about that.
No, you could feel free to chime in anytime. I'm gonna move on from this for the sake of time. So these are the consensus sequences that were made for the samples. I'm just gonna click through those so the students can actually see these. They could copy and paste from here and do what they might want to, maybe put them in BLAST, although we're gonna do BLAST right now. I actually preloaded because BLAST takes a little bit, but we could BLAST all of these to identify what they are. I already did that here, so this should be all ready. But all I did was select them and then BLAST. So I'm just going to go through what the results would look like. So these are the BLAST results. We see the number of mismatches. We see alignment length, e-value. So it's a learning tool to talk about BLAST. I'm just going to quickly look at each one. So this is a fly, and this is, there are links to the Encyclopedia of Life. This will take you to see some more information. And I do want to just point out that this fly is from the family Pyophilidae. That's going to be important when it comes to the tree. All right, we have another sample. You can look at that. And this fly is also from the, genus Pyophilidae, but it's a different species of fly, the waltzing fly. Gonna look at the next sample. And this is a beetle. And this was the smoking gun for the students. So this was how they were able to determine that it was the cadaver was at a very late stage of decomposition. But I just wanted to have that information out there. So when we look at the tree, it makes a little bit more sense. And then I'll quickly look at these. I think this actually was a lower quality run, which is why there's so many mismatches. But this is also a fly, like the first two from the family, genus Califoridae, and this fly is also from Califoridae. Okay, so those are our insects that we were able to identify. Next, we want to do a multiple sequence alignment, which we will need for the phylogenetic trees to understand the evolutionary relationships. So Dave showed some of the examples of these in the very beginning with student projects. So the students are able to look at the alignment and scroll. We know that barcode 6 is a beetle, so you can see that that sequence, if you just look, scrolling tends to be a lot different. But the students are able to explore this alignment and also trim the alignment, which will trim the ends. And then lastly, we have 2 different algorithms to make phylogenetic trees, the neighbor joining algorithm and the maximum likelihood. I'm gonna quickly do both. So I'm gonna choose the beetle as an outgroup because that's the most distantly related to the flies. And these flies, these were the 2 flies that are in the family Pyophilidae, and these were in Califoridae. So it would make sense that they cluster. And our beetle is our outgroup there. And with the maximum likelihood, it is a probabilistic model, and we have, oh, did I pick the right one? Yeah, this is a little bit different. So I do know this sample was a little low quality, which I think is why it's being chucked out there. But okay, so I'm gonna finish this. So this was just a demo, and I know that we're a little low on time. I'm gonna briefly just bring it back to some announcements because I want to make sure that there's time for questions. Okay. Is it showing the PowerPoint?
Yes,
Okay.
that's the PowerPoint.
Okay. So, 2 last things I wanted to mention is that Oxford Nanopore has an education, a separate store for educators that has a special pricing. So there are much lower prices for chemistry kits on this education store. This is information that we just got. This will be the education prices for some of the kits. So the rapid sequencing kit, for instance, is what we use for the PCR products for DNA barcoding, but it's also what we use for some of the genomic sequencing as well. So this is just an idea of the pricing. And we are going to have a series of workshops this summer, educator workshops for using Nanopore in the classroom. And these are the dates that we have right now for those. This link, these aren't live yet, they will be shortly, but this link will bring you to where you can register once these officially go up. Okay, that's all I had to share. Thank you.
Thank you, Dave and Anna. We have about 5 minutes for a Q&A, and at this time I see Keith's hand that was raised. Keith, go ahead. You're on mute, Keith.
Are we talking about to me?
Keith.
Yeah,
Keith had his hand up unless it was an error.
okay.
Oh, okay. Oh, I'm sorry. That was an
Disappointed.
error. You were applauding.
There is a question, have we thought about doing virtual training? We did a ton of it during COVID. We actually, that's how we really did a lot of meta barcode training using Illumina technology. So yeah, I think we'll probably do it, but for this summer, we're having these 5 different ones just because there is some hands-on work and we do like to get personal. But I think we'll probably will do some purely virtual stuff as well. That's a great question.
Mark,
Maggie? Oh, sorry. Oh, you're on mute.
Anna and Dave, really great talk. Thank you. I was wondering about the sort of training workshops that you're going to run in the summer. Are there faculty stipends for community college faculty to attend? Is there help with travel and so on for them?
Yes, I should remember what they are, what the stipends are, but yes, all the teachers who participate will get a stipend. I think it's $300 or $600, or no, maybe it's 500. Anyways, some hundreds of dollars. I should remember. And we do have funding to do travel for a subset of people at each workshop, around about 8 or 10 people per workshop. But we have tried to site most of them in major cities so that we can get a large number of commuters. And that's the way we've been doing workshop for decades because it's pretty cost effective to have a distributed network of sites. But yes, Maggie, we will be supporting the teachers. Last summer, we had just about twice as many teachers apply as we had spaces. So if you know people are interested, have them apply, rapidly, but also make sure they fill out the application fully because we do evaluate that. And as you can imagine, some people are a little more qualified than others. And especially in this day and age, we sure as heck better just take the qualified ones, or it's possible we might go to the guillotine.
Thank you.
Celeste, don't you dare laugh. In Washington, D.C., they got that knife right over your head, I'm sure.
I see a question from Stacy about a workshop in the Northeast. I think this summer Chicago is going to be the closest location to the Northeast.
Anna, there is a question from Scott in the chat about the link for educator ordering from an Annapurna.
I see. Yeah, I can send that out. I don't have it on hand. Is there a way to send Dion like an email to folks for that attend? Is that
Yes, we can. I can get the emails for everyone.
okay? That'd be great. Yeah, because I need to ask the director of education what the best
Yes, we
link
can
is for
make
that.
sure to include that on the website as well.
Okay, thank you.
We have about 1 more minute for Q&A.
Well, I want to thank everybody for tuning in. It's always a pleasure to be with a bunch of teachers on a Friday afternoon.
Thanks so much, everyone.
And just to let you know, I'm sure you saw Joseph also had his whole class listening in. But thank you again, Dave and Anna, for taking time to share your presentation with us. It was incredibly informative and engaging, and I'm certain I speak for everyone in saying that we truly appreciate the insights you provided. As mentioned earlier, the recording for this project talk will be on the InnovATEBIO website, as well as additional information for this ATE project and future ATE projects talks scheduled through the end of May. On behalf of the entire Innovate Bio leadership team, thank you all for joining us and best wishes for a peaceful weekend. Safe travels to all who are traveling today. Bye.